When you're working with microorganisms, there are certain guidelines to follow to make your work safe. Although many species of fungi and bacteria represent minimal risk when handled correctly, it's a good idea to treat all cultures as if they were potentially harmful. Before starting work, wipe down the bench surface with disinfectant, for example, Milton, a hypochlorite solution. This reduces the number of microbes, though it doesn't sterilize. The equipment you'll need: a Bunsen burner, an inoculating loop, straight wire, a glass spreader and a beaker of alcohol, which is used to sterilize the spreader and is kept well away from the Bunsen. It's also useful to have a container holding a disinfectant solution, like Clearasol, onto which you can put used pipettes and cotton wool. They can then be disposed of safely, and contamination and clutter are reduced. When you're removing bulbs from pipettes, make sure that the tip is pointing into the disinfectant to avoid spraying culture into the air. Autoclaving is the most common method of sterilizing apparatus before and after use. The principle behind autoclaving is that water boils above its normal boiling point when air pressure is increased. For example, at 15 pounds of pressure per square inch, water reaches 121 degrees Celsius. If that temperature is held for 15 minutes, both microbes and their spores will be destroyed, and media and apparatus inside the autoclave will become sterile. Aseptic techniques prevent benchtops, apparatus and people from being contaminated by the microorganisms being used. And they also prevent cultures from being contaminated by microorganisms in the environment. Throughout this video, you'll see certain procedures being repeated again and again. These are fundamental aspects of aseptic technique, and they must be performed if your work is to be safe and successful. Flaming is simply heating equipment in a Bunsen flame. Loops, wires, spreaders and pipettes are all flamed in order to sterilize them, but the techniques differ. Loops and wires are flamed by heating them in the Bunsen flame until they glow red. The loop or wire is held almost vertically, pointing downwards, in a roaring flame, and the whole length is heated, then allowed to cool. To avoid contamination, the loop or the wire should never be cooled, resting on the work surface. It's important that loops and wires should be allowed to cool before being placed into cultures. If they're too hot and dipped into broth, an aerosol laden with microorganisms will be produced, whilst organisms on the loop will be killed by the heat. Aerosols are a major source of contamination. They're potentially hazardous as they're virtually invisible, they remain suspended in the air for some time, and they may be inhaled. Similarly, agar in plates will spit if it's touched with a loop which is too hot. After inoculation, the loop or the wire should be sterilized by introducing it into the Bunsen flame, but slowly, to avoid further aerosols. Spreaders are sterilized using alcohol, not by heating in a Bunsen flame, since this might cause the glass to snap, and the temperature of the glass would instantly fry any microorganisms and cause aerosols. The spreaders removed from the beaker and passed through the Bunsen flame to ignite the alcohol. This heats the spreader to about 60 degrees Celsius, which is enough to sterilize it, but not hot enough to break the glass. The spreader cools almost immediately. After use, it should be put back in the beaker. Bottlenecks are also flamed, not to sterilize them, but to create a convection current in the neck of the bottle, which lifts the air out of the bottleneck and removes any microorganisms suspended in the air around the top of the bottle. This helps reduce contamination. The bottle doesn't have to stay in the flame till it glows. 2 or 3 seconds is enough. The uplift of air carrying microorganisms away from the immediate area is another reason why all microbiological work is carried out near a Bunsen with a roaring flame. It helps to prevent aerial contaminants from falling onto media. Return the Bunsen flame to yellow when you're not using it. It's easier to see the flame when it's yellow than when it's roaring. Bottle tops are held between the little finger and the palm of the pipette or loop hand. Never put them on a bench, since they might be contaminated. Holding caps in the little finger also allows them to be replaced easily. It's difficult at first, but it becomes easy with practice. It's a good idea to loosen bottle caps before work starts. This makes handling the bottles easier when carrying out aseptic technique. Pipettes are flamed by flashing them through the Bunsen flame in one continuous movement. Most microbiological work is carried out using Petri dishes containing sterile solid medium. These are known as plates. They're made simply by taking a sterile Petri dish and pouring in about 15 cubic centimetres of fresh sterile medium from a McCartney bottle. The plate is then swirled to ensure even coverage of the surface. Once the medium has set, the plate's ready to use. All dishes should be labelled with your name, the date, the name of the organism, or the source of the organisms. The base is labelled in preference to the lid because if the two halves become separated, the growth on the medium can still be identified. Plates are always dried with the base inverted and resting on the lid to reduce the risk of contamination. Plates can be dried in an incubator, but it's also okay to dry them on a bench where they won't be moved. When plates have been inoculated, they should be sealed with adhesive tape. Don't seal them completely, as this may encourage the growth of potentially harmful anaerobic bacteria. Cultures are incubated to allow the microorganisms to multiply and grow, resulting in the formation of visible colonies on agar plates or turbidity or cloudiness in broths. The optimum incubation temperatures vary depending on the organisms, but it's generally agreed that in schools, 27 degrees Celsius is a suitable temperature for the incubation of bacterial cultures, whilst fungi should be incubated at 20 degrees. Bacteria are generally incubated for 24 to 48 hours, whilst fungi are incubated for 4 to 5 days. Culture shouldn't be incubated above 30 degrees, as this might encourage the growth of organisms capable of causing disease in humans. These organisms often have an optimum growth temperature of 37 degrees Celsius. The substance used for the growth of bacteria and fungi in the lab is known as the medium. Media can be either solid or liquid, in which case it's known as a broth. There's no nutritional difference between solid and liquid media. Both contain all the necessary requirements for microbial growth. Solid media, however, contain agar as a solidifying agent. Agar is obtained from a type of seaweed and is of no nutritional value, but it does have unique properties as a solidifying agent because it melts at 100 degrees Celsius and it sets at 40 degrees Celsius. It can be maintained in a molten state in a 50 degrees Celsius water bath. Agar will remain liquid over a range of temperatures and it won't set too quickly, giving time for it to be used. If microorganisms are added to molten agar, for example in pour plates, the agar shouldn't be over 45 degrees Celsius. The bottle will be hot enough to hold in your hand without being uncomfortable. If it's too hot, the microorganisms are killed. If it's too cold, the agar will set and it'll need heating to 100 degrees Celsius again to remelt it. There are many different types of media containing many different combinations of nutrients, but a good medium for general bacterial work is nutrient agar. Fungi have different nutritional requirements from bacteria, and so nutrient agar isn't the best medium for them, although they will appear as contaminants quite readily. When your experiments are finished, clean the work area thoroughly and dispose of used cultures, Petri dishes and other equipment carefully. Used Petri dishes and cultures must either be placed in a solution of suitable freshly diluted disinfectant or placed into autoclavable plastic bags and autoclaved before disposal. Pipettes and cotton buds are placed into freshly diluted hypochlorite solution. The bench should be wiped down thoroughly with disinfectant or decontamination fluid to kill any microorganisms which may have been spilt. Finally, wash your hands thoroughly before leaving the lab. Streak plates are used to isolate bacterial or yeast colonies. One well-isolated colony has usually grown from one bacterial cell originally deposited on the agar. It can be assumed that all cells in the colony are the same and the colony is pure. The isolated colonies can be used to inoculate fresh, sterile agar plates on which pure cultures will then grow. The properties of organisms in pure cultures can then be studied, unobscured by contaminants. Streaks are made using a wire loop on sterile agar medium in a Petri dish. First, the loop is sterilized by heating and then allowed to cool. The top of the culture bottle is removed. Notice how the top and the loop are held. The neck of the bottle is then flamed. The loop is placed into the culture bottle and a loop full of suspension is taken out. The neck of the bottle is reflamed and the top replaced. The lid of the Petri dish is raised and the first sector of the plate is streaked gently by drawing the loop across the surface of the agar. It's important not to press down on the surface of the medium, as this can cause gouging and possibly unintentional growth of anaerobes below the surface of the agar. After streaking the first area of the plate, the lid should be replaced and the loop flamed and allowed to cool. The second sector of the plate should now be streaked by drawing some of the original inoculum from the first sector with the sterilized loop. Again, the lid is replaced and the loop is flamed and cooled before streaking a third area of the plate, drawing from the second streak. After flaming the loop yet again, the final streak is taken by drawing from the third streak to the center of the plate. The loop is flamed once more. The plates are inverted and incubated at 27 degrees Celsius. If the streaking has been successful, colonies should be clearly visible on the plate after incubation. Growth is confluent around the initial streaks. The colonies grow and merge. The aim is to deposit most of the bacteria in the first few streaks. Subsequent streaking has a diluting effect, producing isolated colonies. The pour plate technique can also be used to isolate single colonies for pure cultures. It's easier to perform than streaking, and it consists of inoculating the culture into a sterile tube or bottle containing molten agar at 45 degrees Celsius. The suspension can't be too concentrated, otherwise confluent growth will be obtained rather than isolated colonies. The culture bottle is opened and flamed, and between 0.1 and 1 centimetre cubed is removed using a pipette. Notice the cap is held in the little finger of the pipette hand. The culture bottle is then reflamed and the top is replaced. The medium bottle is then opened and flamed and inoculated with the mixed culture. The bottle is reflamed, the top replaced, and the pipette disposed of carefully. Then, to disperse the bacteria throughout the medium, the bottle is gently rotated in the palms. This prevents bubbles developing in the agar, which would be transferred onto the plates and which would then set. The mixture is poured onto a sterile Petri dish Notice that the lid of the Petri dish is never completely removed. Once the medium has set, the plate can be inverted and incubated. Alternatively, the inoculum can be placed into an empty Petri dish and the melted medium poured over it. The dish is swirled gently with fingertips on the lip to mix the medium and the bacteria before setting. Colony growth takes place within the medium and on its top and bottom surfaces. As each colony develops from a single bacterium present in the original inoculum, this method can be used for counting the number of bacteria present in a sample of liquid, as long as you know the dilution factor. A sheet of bacterial growth over the surface of a medium results in a bacterial lawn or confluent plates. Such plates can be used to assess the sensitivity of a microorganism to various reagents, for example, antibiotics, or even toothpaste or mouthwash. Lawns can be prepared in one of two ways. Firstly, the lawn can be created by simply flooding a sterile agar plate. To do this aseptically, the culture bottle is opened and the neck flamed. The lid of the Petri dish is then raised, and enough bacterial culture to cover the surface of the agar is tipped into the plate, about 1 cubic centimetre. The plate is then swirled, allowing the culture to cover the surface of the agar completely and to distribute the bacteria evenly. Any excess culture is discarded by tipping it into a solution of disinfectant. The plate should then be left to dry before incubation. A safer way to prepare a bacterial lawn is the spread plate method. This involves pipetting between 1 and 2/10 of a cubic centimetre of bacterial culture into the centre of a sterile plate. The bacteria are then evenly distributed over the surface of the agar using a glass spreader, sterilised by flaming in alcohol. The lid of the culture bottle is removed and the neck of the bottle is flamed. Using a sterile pipette, 0.1 to 0.2 centimetres cubed of the bacterial broth is removed. The neck of the culture bottle is then reflamed and the lid is replaced. With the lid of the Petri dish raised slightly, the culture is pipetted into the centre of the plate and the pipette then discarded. The glass spreader is then removed from the alcohol and flamed to sterilise it. Obviously, to avoid the risk of fire, the beaker containing the alcohol shouldn't be too near the Bunsen flame. When it's cooled, the spreader can then be placed lightly on the surface of the agar. The dish should be rotated whilst the spreader is moved up and down and across the medium. Once the lawn has dried, reagents can be added to measure inhibition of growth. If the reagent to be tested is solid, for example toothpaste, it can simply be placed in the middle of the plate. If liquids are to be tested, wells must be cut into the medium using a cork borer, sterilised by flaming in alcohol. It's important that the borer is held horizontally in the flame to prevent hot gas passing up through the centre of the borer and burning your hand. Remove the agar plug using a flamed wire if it doesn't come out when the borer is withdrawn. When the well has been cut, the liquid can carefully be pipetted into it using a sterile Pasteur pipette. Before cutting wells, make sure the medium has set completely. This makes it much easier to cut smoothly. Inhibition of growth is demonstrated by clear zones around the well. Inhibition due to antibiotics is demonstrated by placing paper discs containing antibiotic onto the surface of the agar. Multi-discs can also be used to show inhibition of bacterial growth due to the presence of antibiotics. These are simply filter paper discs with a number of different antibiotics placed on spokes around the edge of the disc. These are placed onto a bacterial lawn, and again, inhibition by antibiotics will be indicated by a clear zone. Multi-discs are useful as they allow the testing of more than one antibiotic for antimicrobial properties. Slopes are a useful way of maintaining cultures. The slope is made by allowing melted agar in a McCartney bottle to set whilst the bottle is on an incline. The slope shouldn't reach more than 2/3 the length of the bottle. Slopes give a large surface area to a small volume of medium, usually 15 cubic centimetres. In other words, they maximise the area available for bacterial growth. Slopes are usually inoculated with the chosen organism using a wire loop. The loop is flamed and allowed to cool. The top of the culture bottle is removed, the neck flamed, and a loop full of broth removed. The bottle neck is then reflamed and the top replaced. The slope bottle is then opened, flamed, and inoculated by drawing the loop across the surface of the agar, starting from the bottom of the slope, moving the loop from side to side towards the top. Once that's done, the bottle neck should be flamed and the lid replaced. Finally, the loop should be flamed to sterilize it. Stabs, or deeps as they're sometimes known, are used to culture anaerobic bacteria, but they can also be used to separate aerobic and anaerobic bacteria in mixed culture. Whilst this procedure is routinely used in microbiological laboratories, it's not recommended for use in schools. This is because many pathogenic organisms grow successfully in anaerobic environments. With stabs, the medium is inoculated using a straight wire instead of a loop, but other than that, the procedure is identical to that used with slopes. The wire is flamed and allowed to cool. The culture bottle is then opened, the neck flamed, and the wire placed into the broth in order to pick up some of the bacteria. The culture bottle is then reflamed and the lid replaced. The stab bottle is then opened, the neck flamed, and the medium inoculated by pushing the wire smoothly into it to a depth of 3 to 4 centimetres. The bottle should then be reflamed, the top replaced, and the wire flamed to sterilise it. Notice the growth of the anaerobic bacteria occurs only at a depth in the agar where air is excluded. To inoculate an agar plate with fungi, a different technique is used. This is because it's difficult to transfer spores or hyphae with a loop. Spores can be used as sources of inocular, depending on the species and the nature of the investigation. Mycelial discs can be used to inoculate plates. The end of a 5mm cork borer is dipped in 75% alcohol, flamed, and allowed to cool. The Petri dish containing the fungi is opened and the cork borer is pushed into the margin of the fungal colony. This is the region where growth is most vigorous. Behind the margin, growth has slowed or stopped, and the fungi from there are unsuitable for inoculation. If the plug remains in the borer, it can be removed using a flamed wire. The wire should then be flamed to sterilize it. Invert the dish and then incubate at 20 degrees Celsius for 4 days. Alternatively, fungal spores can be used to inoculate a plate. A small amount of material is removed from the mycelium and placed onto the center of a sterile Petri dish containing malt extract agar. The plate is then incubated again at 20 degrees Celsius for 4 days. If you follow the simple instructions on this video, it should ensure that your introduction to practical microbiology is safe and enjoyable.